All right, all right, all right, all righty. So what's going on? Good afternoon to everybody. Good afternoon. This is Tiffany. I'm here live in effect. So today's topic is going to be about Black history because this is Black History Month, and I want to speak on Georgia Dunston, who is a professor at Howard University, and she also a human geneticist. So it's important that we talk about those in STEM, okay? Because there's preconceived notion that there's not black people in the STEM program, which there is, but you have to really look them up. And so I think it's very important to focus on those who are in the genetics field, biology field, and anthropology field as well, because when we talk about human and genetics, right, we really have to be able to get into those components, okay? And we have to really study those who actually done the work. So there's people who might be familiar with Rick Kittles, who's the founder of African Ancestry. Okay, he's the founder of African Ancestry. But there's many lists of people who's in the biology field and anthropology field. So Kieta is a bioanthropologist. Dr. Fatima Justice, she's a bioanthropologist, right? But when I watched this lady here, Georgia Dunston, I said I had to do a video about her because she was on TED Talks, right? Back in, I think it was back in 2016 at Howard University. And she was basically talking about a biological behavior and the genome. And she does her research on that. She gets involved into doing all those different things. So before I get started, please make sure you like and subscribe to the channel. Hit the notification bell. Share the channel. Okay. So I want to go ahead and get started on some information about her, and then I want to get into some journals that she has done, and I want to show you guys the website. You can find out more information on her as well. So let's go down to. So I would go to HistoryMaker.org. Okay. So you can go on HistoryMaker.org website. All right, so Georgia May Dunston, right, who is a geneticist, was born in Norfolk, Virginia on August 4th of 1944 to a working class family. As a child, Dunston developed an interest in biology of race and decided to continue her study of biology after graduating from high school. She earned her BS, which is a Bachelor's of Science in Biology from Norfolk State University in 1965. And she earned her MS degree in biology, which is Master of Science from Tuskegee University in 1967. So Dunston went on to study at the University of Michigan, finishing her PhD degree in human genetics in 1972. She then accepted a position at Howard University Medical Center as an assistant professor, which she held from 1972 to 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1978. 1975 to 1976, Dunston completed a postdoctoral fellowship at the National Cancer Institute where she studied tumor immunology. She later served as a scientist there in an immunodiagnosis lab that was partly funded by the National Institute of Health. At Howard, Dunston was appointed director of the Human Immunogenetics Laboratory in 1985. At this time, she focused her research on diseases that are common in the Black community, as well as genes and immune reaction that are unique to African American population. From 1991 to 1994, Dunston served as Associate Director of the Division of Basic Science at Howard University Cancer Center. She was promoted to full professor in the Department of Microbiology at Howard in 1993 and became chair of the department in 1998. Inspired by the Human Genome Project begun in 1990, Dunston focused her attention on the genetic heritage of the African American population. Dunston worked in human genetics and diversity resulted in her founding the National Human Genome Center at Howard in 2001. So it goes on to say Dunston is the recipient of several awards, including the Howard University College of Medicine Outstanding Research Award, NAACP Science Achievement Award, the Howard University Graduate School of Arts and Science Outstanding Graduate Faculty Member Award. She has been a member of the National Advisory Council for the National Institute of Environmental Health Sciences, Sigma SI, and the National Academy of Science Review Committee on Human Genome Diversity Project. So she currently lives in Washington, D.C. So again, this comes from the HistoryMakers.org, okay, which is dealing with the website about Black people in history. They do interviews with those who are in the field of fine arts, also those who are in the field of science, music, entertainment, etc. Okay. All right, so that's that information. Let's see what else we can find. All right, so that's all the information. Now, I want to also show you guys this. If you guys are interested, this website is National Human Genome Research Institute. Okay, the genome.gov. So this is the interview that she done. You can check it out on YouTube. So it says she is the founder of Whole Genome Science Foundation, a nonprofit to advance knowledge on the science, technology, engineering, and mathematics of the human genome. She is also the founder of former director of the National Human Genome Center at Howard University, where she also taught human immunogenetics. In part one of this oral history, Georgia talks about her passion to understand humans from an early age and how her interest in human genetics led her to study biology at Tuskegee University and human genetics at University of Michigan and Harvard. All right. So then she discusses her time in the visiting investigators program at NHGRI and how that led to collaborating with Francis Collins on publishing work on the genetics of type 2 diabetes in West Africa. Okay. So it's so many lists of things on here, okay? You have many people that are in the scientific field that has been interviewed, but again, you can go to this website and you can check out her interview here, right? So she has a lot of interesting stuff. So I'm gonna go I'm gonna go into details with some of these articles. All right, so the first article she did is the one about a genome-wide association study on African ancestry population for asthma. So it's multiple people that was involved in this, but she happened to be one of those people that was involved in this study as well. Okay. Oh, my voice. All right, so it says background asthma is a complex disease characterized by striking ethnic disparity, not explained entirely by environmental, social, cultural, or economic factors of the limited genetic studies performed on population of African descent. Notable differences in susceptibility allele frequency have been observed. All right, so they go into the introduction and they go into the methods. All right, so they talk about the research and studies they did on the study of African Americans and asthma. So it says introduction. Asthma is a complex disease characterized by intermittent inflammation of the airways. Morbidity and mortality rates are disproportionately high among ethnic minorities, including African American. They continue to rise. The striking ethnic disparity in asthma prevalence cannot be explained entirely by environmental, social, cultural, or economic factors. Nearly a dozen genome-wide linkage screens and two recent genome-wide association studies have confirmed a strong genetic components to asthma. It remains difficult, however, to identify specific causal genes and determine whether genetic control contributes to the observed ethnic disparity for this complex disease. In this study, two independent populations of African descent asserting through physician diagnosed asthma have been recruited by a consortium entitled Genome Research on Asthma in the African Diaspora. These populations have been genotyped using the Illumina human HAP 65OY B chip containing 655,352 SNPs as part of a genome-wide search for genes controlling risk to asthma in ethnic minorities. So it said generalized ability of findings from these population of African descent was tested in European family and case control panels of UK and German origin, respectively. For example, of African Americans from independent. So let's go into the European ancestry replication sample. So in addition, we also use data from an earlier genome-wide association study for childhood asthma in Caucasian sample described elsewhere. So briefly, this study involved family and case control panel comprised 994 patients with childhood onset asthma and 1,243 non-asthmatic. All right. So then it goes down to say that the family panel consists of 270 predominantly nuclear families asserted through a proband with severe childhood onset asthma. These families contain 295 SIP bars, 11 half SIP bars, and 3 singletons. In addition, a set of 437 non-asthmatic age matched Caucasian UK controls were also studied. So the case control panel consists of 728 asthmatic children from the MAGIC studies and 694 matched non-asthmatic children recruited by the Isaac study. Also, all cases in both family and case control panels had physician diagnosed asthma. All right, so then it goes down to talk about African American replication sample. So CHOP for replication of findings in one of the studies with existing GWAS African Americans children were recruited at Children's Hospital of Philadelphia, which is CHOP, between 2006 and 2008. Case includes 1,456 patients with physician diagnosed persistent asthma. Control include 1,973 subjects who were determined to have no history of asthma or reactive airway disease by questionnaire and who had never been prescribed asthma medication according to their medical records. Mean ages of case was 7.5 to 5.7 SD years, and 57% were male. Mean age of control was 6.7, 5.2 SD years, and 49% males. Okay. So let's go further down. So again, goes into the methods and how they went about studying, etc. So I want to skip all of this. So I'm gonna go into the results. All right, so it says administrative analysis revealed ancestry misclassification for 7 individuals among the African American subjects and 18 individuals from ethnically mixed family from Barbados who were also excluded from subsequent analysis. Additionally, samples were dropped based on quality control, analysis of familial relationship, and Mendelian inconsistency. 14 sample in African Americans groups and 1 in the African Caribbean group revealed gender discrepancies compared with clinical records among all African American cases, and control combined 27 individuals were dropped because identity by state estimated suggests duplicated sample. So 29 pairs of individual had estimated IBS, which is identity by state, equals 0.50, suggesting they were first degree relatives, but were retained for analysis, resulting in a total of 464 asthma cases and 471 non-asthmatic control. In the families from Barbados, 26 pairs of duplicate sample were identified, and 13 individuals had greater than 1% of available markers showing Mendelian inconsistency, suggesting a biological relationship different from the reported family structure. These individuals were dropped, resulting in a total of 929 subjects from 163 pedigrees in the final family sample from Barbados. All right, so basically they was going over the studies about African Americans and the history of asthma. All right. So again, that website, if you are interested, is called National Library of Medicine, National Center for Biotechnology Information. All right. So going on to the next one. This one deals with the assembly of a pan genome from deep sequencing of 910 humans of African descent. All right. So this came out November 19th of 2018. Right. Okay. So I'm just showing the journals, the article that she did, etc. Just walking through it, whatnot. Hmm. Let's see something. All right, so let's go into the abstract. So it says, we use a deeply subsequent data set of 910 individuals of all African descent to construct a set of DNA sequence that is present in these individuals but missing from the reference human genome. We align 1.19 trillion reads from the 910 individuals to the reference genome, collected all reads that failed to align, and assemble these reads into contagious sequence. We then compare all contents to one another to identify a set of unique uh, sequences representing regions of the African pan genome missing from the reference genome. Our analysis revealed 296,400 no 296,485,284 BP in 125,715 distinct contents present in the population of African descent, demonstrating that the human pan genome contains 10% more DNA than the current human reference genome. And although the functional significance of nearly all of this sequence is unknown. 387 of the novel contents fall within 315 distinct protein coding genes, and the rest appear to be intergenic. All right, so not to go into all of this, but just to show what the studies was about and what her avenue is. So basically, it deals with the pangenome from sequence of, basically deals with like the component of the genetics and whatnot. Um, so if you guys want to look into this, you guys can read into this as well. Okay. Now, I have the last article here that I want to show y'all. Let me see. Okay, boom. So now this one deals with a continuum of admixture in the Western Hemisphere revealed by the African diaspora genome, which came out on October 11th of 2016. Okay. All right, so that was one of her articles. So the abstract is the African diaspora in the Western Hemisphere represent one of the largest forced migration in the history and had a profound impact on the genetic diversity in modern population. To date, the fine-scale population structure of the descendants of African diaspora remain largely uncharacterized. Here we variation and suggesting much variation remains to be discovered in African admixed population in the Americas. We summarize genetic variation in these populations, quantifying the post-colonial sex bias European gene flow across multiple regions. Moreover, we refine estimates of the burden of the deleterious variants carried across population and how this varies with African ancestry. Our data are an important resource for empowering disease mapping studies in African admixed individuals and will facilitate gene discovery for disease disproportionately affecting individuals of African ancestry. All right. And it says, here's the introduction: a disproportionate burden of mobility, disability, and death from common chronic disease associated with modern lifestyles persists among U.S. racial and ethnic minority population, most notably among individual African ancestry. Unfortunately, the complexity of colonial history has been highly understudied and homogenized. African Americans and admixed population in Latin America and the Caribbean are grouped into a single racial construct by the American census, which then is often applied in studies of health disparity. This fails to capture the distribution of genetic variation among these populations. In medical genetics, this is especially problematic where studies of population of African descent in the Americas do not adequately account for fine scales population structure resulting from the components of congenital ancestry, and particularly the lower linkage disequilibrium and decreased genome-wide array coverage in these populations. Although the Americas have been a source of large genome-wide association study, population of African descent continue to be understudied. All right, so then it goes on to say to address issue of genome-wide genetic diversity, ancestry, admixture, and limitation in commercial genome-wide association studies array coverage in population of African descent in the Americas representing the African diaspora. We performed the largest whole genome sequencing study to date on population with African ancestry in the Americas. We sequenced 642 unrelated individuals who self-reported African ancestry from 15 North America, Central and South America and Caribbean population, plus Yoruba-speaking individuals from Ibadan, Nigeria, as part of the consortium on asthma among African ancestry population in the Americas. So it says these data substantially increase the lexicon of known human genetic variation and suggest an abundance of variation remains to be discovered with more studies of African admixed population in the Americas. We summarize genetic variation resulting from the African diaspora across the Africas and into the Caribbean, quantifying the post-colonial sex bias European gene flow across multiple regions. Moreover, leveraging our high coverage whole genome data, we are able to refine estimate on the burden of the deleterious variant carried across population and how this varies with African ancestry. Our data will serve as an important resource for empowering disease mapping studies in African admits individual and facilitate gene discovery for diseases disproportionately affecting individuals of African ancestry. All right, so it deals with the result study design overview. Okay, so as we can see, let me see if I can tap on this picture. No, I can't tap this picture. All right, so here it is. Here's a study of people from African descent, right, and European descent. So it's not too much of Native American descent, right? But this is the study they did as far as the admits population is concerned, etc. Okay. Okay, so as you can see, this is the diagram at the bottom. Okay. All right, so right down here says the geographic location of the 15 North and Central and South American Caribbean population sequence for this study are illustrated in Figure 1A, and detailed information on each of the 15 individual component population described in detail. Supplementary Note 1, Supplementary Note 2, 2-9, Supplementary Figs 1 through 3, Supplementary Tables 1 and 2 contain detailed information on subsequent and quality control pipelines, asthma status, ancestry, observed genetic variation, sequencing depth, and calling. Let me see something. Okay, trying to see what I was at. All right, so although designed as a case control study for asthma and associated with phenotype, this systematic characterization of ancestry in all individuals had merit on its own. Asthma is a disease of moderate heritability, yet few loci have been discovered in population of African descent. And contrasting the study site yields large social, cultural, and environmental heterogeneity that could affect asthma risk. Therefore, across the entire genome, patterns of variation would tend to receive little confounding from our case control design. All right. So again, that's just the general idea of what she was into and what she was studying. So I'm not a geneticist, all right, and I don't, I'm not an expert on that. However, that's just basically just give you an idea of what her field of study is. So by her being a human geneticist, right, she studied genome, she studied inheritance, like as far as like what type of disease that you inherit. Like, do you inherit cancer? Do you inherit diabetes, blood pressure, asthma, all of that? She also deals with epigenetics. And one of the most common thing in our community, of course, is not only high blood pressure and diabetes, but we also have a commonality of sickle cells. All right. And we have lactose intolerance. So we know that us as African descended people in America, we inherit that from our African ancestors because they were lactose intolerant and they had sickle cells anemia, to protect them from malaria disease. Okay. And malaria was brought here in America. So hopefully I can get somebody one day to have that conversation with about malaria and sickle cell and expound more on that, because we really need to be talking about this. We really need to have this important conversation dealing with our health. All right. But that's one of the things that geneticists deal with. They study about all of that, and then they study about the ancestry. They study about your bloodline, your ethnic group, everything. So that's really important. So when people start talking about white folks have old blood type and recessive genes, and black people have dominant genes, well, then you have to ask yourself, well, what's your blood type then to make you dominant? And how do you know that you don't have recessive gene? Because we do carry European ancestry in us. So we have to ask ourselves, well, how do we know that we don't have recessive genes? How do we know our blood type? How we know that our blood type is not dominant? Because just because we African descendant people, we don't carry one particular blood type. We have various different blood types out there, and based on your blood type, it will tell a lot about what you're going to inherit and what you're going to deal with as far as your health disparity is concerned, and all of that. So I just wanted to put that out there. But anyways, I hope you guys are having a great one. Y'all be safe. And until next time, may peace and power elevation be to all of you. This your girl Tiffany. I'm logging off. Thank you.